And the answer is that meiosis occurred in the child's mother. That's the one place where a chromosome from the grandmother and a chromosome from the grandmother were both present, as shown here. So it's in the mother that there was the meiosis that produced the mother's egg had a crossover at this position, and that crossover generated this recombinant chromosome that was put into the egg and then gave rise to the child. Now, here's a more complicated diagram. It goes back another generation. So we've now got 4 generations that have been typed by 23andMe. Again, the light blue line is continuous for the length of the chromosome, and that shows identity by descent between the mother and the child, exactly what we would expect. The green segments show identity between the child and their grandparent. So these are segments of the grandparent DNA. Here I've shown it as if it was the grandmother, that were passed by the mother onto the child. And again, the breakpoints between the green segments and the not green adjacent segments represent crossovers that happened in the mother between the green chromosome and the white chromosome, the grandparental chromosomes. Finally, the dark blue lines represent segments that the child inherited intact from the great-grandfather. And you can see that overall there is only about half as many segments that are dark blue as there are segments that are green. Now, in this diagram, you should be able to identify segments that have the properties that indicate the events that gave rise to them. So there may be segments, here's an example of one, where the great-grandparental segment was inherited intact in the child. Nothing changed in the grandparent. So that's a place where crossovers happened outside of this segment. No crossovers happened in this segment. You can also find segments where the great-grandparental segment is shorter at one end than the grandparent, so there would have been a crossover there, and segments where the great-grandparental segment is shorter at both ends here, and we can say, well, there would have been a crossover there and a crossover there. Finally, we can find segments that did not come from the great-grandparent at all, such as this one. There's no dark blue DNA in this region at all. This would be a segment that came from the other great-grandparent. And we can find segments where there's no segment even from the grandparent. For instance, this whole chromosome, the child inherited the grandmother's chromosome and nothing from the grandfather. Now I'm going to show you one more comparison, and that's between 2 siblings. And even though 2 siblings have the same 50% identity predicted as a parent and a child, the pattern of sequence identity is very different. Between parent and child, it's one allele is identical throughout the whole length of all the chromosomes. But for siblings, there are segments indicated by dark blue where the 2 siblings have identical alleles at both alleles. So they both got the same allele from their mom, and they both got the same allele from their dad. There are lots of segments indicated in light blue where they're half identical. That means they either both got the same allele from their mom, or they both got the same allele from their dad, but not both. And then there are long white segments where they got different alleles from their mom and different alleles from their dad. So there could be parts of your genome where you're identical to your sister, and other parts where your genomes are completely different. So what we've done, we talked about paternity testing, how to think about DNA for fingerprinting in the context of parent-child relationships, you can also extend this, of course, to thinking about more distant relationships. For instance, for immigration hearings, it's often desirable to have evidence of a family relationship. For instance, if you wanted to be able to bring your nephew into the country, you might be asked to provide DNA fingerprinting type evidence to show that this person actually is your nephew. We considered the average predicted levels of relatedness for different kinds of family relationships, and then we looked at what real examples of relatedness are like from real SNP typing data. Coming up next, we're going to continue thinking about relatedness, but we're going to move farther back in time and think about what happens to ancestral haplotypes, segments of DNA that we inherit from more distant ancestors. I hope to see you there.